Hello and welcome to MLE Masters, your free YouTube learning series created to support students who are struggling with their studies. In today's video, we're going to explore one of the most important biology topics, the cell cycle and how it's regulated. We begin with the G1 phase. This is the first stage of the cell cycle, where the cell undergoes growth and prepares for DNA replication. It's also the stage where DNA repair happens, particularly through nucleotide excision repair. Sometimes the cell exits G1 and enters a resting phase called G0 or G0. In this phase, the cell is not dividing but continues performing its normal cellular functions. Although it looks small in diagrams, this phase is critically important, as many working cells in the body stay here. If the cell continues in the cycle, it enters the S phase, also called the synthesis phase. This is where DNA replication occurs. The amount of DNA doubles, but the number of chromosomes remains the same. The cell is now preparing itself for division. After the S phase, the cell enters G2. Here it undergoes more growth and makes final preparations for mitosis. The cell checks whether everything is in order before it proceeds to divide. The M phase stands for mitosis, the actual division phase. The cell divides into two identical daughter cells, each with the same DNA content. After mitosis, cells either go back into the cycle or enter the G0 phase, depending on what the body needs. The cell cycle is tightly regulated by proteins called cyclin-dependent kinases, or CDKs. CDKs are enzymes that activate other proteins by adding phosphate groups, but they only work when they bind to another protein called cyclin. Cyclin production is triggered by external growth signals. When cyclins bind to CDKs, they form an active complex. For example, cyclin D binds to CDK4, and together they help the cell move forward by activating specific target proteins. Move on to the checkpoints. The most important checkpoint in the cycle is between G1 and S phase. This checkpoint is controlled by cyclin D and CDK4. It's also regulated by two important tumor suppressors, P53 and RB, which play a key role in preventing uncontrolled growth. The next checkpoint is between G2 and M phase. It ensures that all the DNA has been correctly and completely replicated. If there's any damage, the cycle pauses to allow repair. If the damage cannot be fixed, the cell may undergo programmed death. During mitosis, there's one last checkpoint, called the spindle checkpoint. This ensures that all chromosomes are properly attached to spindle fibers. If not, the cell will not proceed with division and may initiate apoptosis to prevent errors. Let's take a closer look at P53 and RB. The RB protein binds to a transcription factor called E2F and prevents the cell from progressing. When CDK4 and cyclin D phosphorylate RB, it releases E2F and allows the cycle to continue. If there's DNA damage, the protein P53 is activated. Known as the guardian of the genome, P53 activates another protein called P21, which stops CDKs. This keeps RB bound to E2F and halts the cycle. The cell then either repairs the damage or undergoes apoptosis if repair is impossible. When DNA damage is severe, P53 activates BAX and BAC. These proteins puncture the mitochondrial membrane, releasing cytochrome C into the cytoplasm. Cytochrome C then binds APF1 to form the apoptosis, which activates caspase 9. This triggers a cascade that activates caspase 3 and caspase 7, breaking down the cell and leading to apoptosis. This elegant process ensures that damaged cells are removed without harming the surrounding tissue. Let's summarize everything we've learned. G1 is the growth and repair phase. G0 is the resting phase where most working cells stay. S phase is where DNA replication happens. G2 is the final preparation phase. M phase is mitosis, where the cell divides. The cycle is regulated by checkpoints and key proteins like CDKs, cyclins, P53, and RB to ensure everything goes smoothly. We hope this video helped you understand the cell cycle in a simple and clear way. If you found it useful, please don't forget to like, share, and subscribe to MLE Masters. We're here to help you learn smarter and succeed in your studies. Thanks for watching, and see you in the next video.